been our companions and protectors for thousands of years. But how much do we really know about man's best friend? Venture into your local dog park on any given day, and it's plain to see the differences in size, shape, and colour among dogs. Today, due to selective breeding by humans, dogs exhibit a wide range of body types. In fact, dogs have the greatest variation in body size and shape of any mammal species. In 2010, the biology article, A Simple Genetic Architecture Underlies Morphological Variations in Dogs, researchers developed a map of canine variation in order to find correlations between gene markers and traits in the breeds. The researchers found that a simple architecture underlines the great variation in domestic dogs. More than 60,000 potential single nucleotide polymorphisms, or SNPs, were genotyped using DNA isolated from 915 dogs, which included 80 domestic dog breeds, 83 wild dogs, and 10 African shelter dogs. Physical measurements from individuals, as well as skeletal measurements from museum specimens, were obtained for a genome-wide association map. The map of phenotypes comprised of 57 traits, including body size and dimensions, cranial and dental shape, and long bone shape. This can be viewed on figure A and B. This map allowed for researchers to identify genomic regions responsible for many key physical characteristics. Examining the map, each row corresponds with a trait, and each column corresponds to a genetic region that has been associated with at least one trait. The shading of each rectangle shows the significant associations. The darker the shading, the greater the association. For most of these traits investigated, it was found that a few QTLs of large effect govern the phenotypic differences among breeds. The SNPs are CFA4.41, CFAX.85, CFA7.46, CFA10.10, CFA15.43, CFAX.85 and CFAX.104. The region on CFA10 is most likely responsible for the difference in ear type. The other regions listed are largely dictated by skull shape. It was also found that the CFAX.104 region that is associated with body size is also associated with skull length. Nearly all these regions were associated with dental traits. The breed mapping reveals that, contrary to wild species, only 6 or 7 locations in the dog genome are necessary to explain about 80% of the difference in height and weight among dog breeds. The simple architecture of phenotype diversity in dog breeds indicates that breed dogs and village dogs went through a bottleneck at domestication, resulting from strong selection as humans selectively bred dogs. The results demonstrate the efficiency of mapping multiple traits in the domestic dog using a database of genotyped individuals, and, in conclusion, highlight the important role human-directed selection has played in altering the genetic architecture of key traits in this important species.